The scientific method is a super important and super exciting part of all different types of science. Science is the investigation and exploration of natural events and of new information that results from those investigations. Science looks different depending on what exactly you're looking at as far as natural events go. If you are looking at the interactions of matter and energy, we're talking about physical science. Those would be the types of things you most likely studied in sixth grade, like forces and chemistry. Life science would be the study of all things that are or were once living. That's what we'll study this year in seventh grade. And earth science is going to consider all different things about the earth and outer space. So we'll look at the weather, plate tectonics, rocks, and astronomy, so looking at space. This year we will focus on life science, the study of living and non-living things. So that's what the rest of this flip will focus on. As you move through this flip, keep in mind that the scientific method is a list of steps or a set of steps that scientists use as they're completing an investigation. They can look and be worded differently, but no matter what words you use, the scientific method still follows this same process. On your flip notes guide, you'll find some blanks with numbers next to them throughout the flip. Each of those blanks consists of or considers one of the different 6 steps that we will talk about in the scientific method. So you may go through and pause right now and add in each of the different steps next to where you find a 1, a 2, a 3, a 4, a 5, and a 6 with lines next to it with the same number of words. Now that you've got it written down, I want to just talk through briefly what the different steps are, and then we'll focus on the first several parts of the scientific method. So as you move through the scientific method, you first ask a question. And when you're asking questions, they result from making observations, inferences about things. They might result because of research. But no matter what's happened, you are going to then make a hypothesis. As you make those observations, do some research, you'll come up with what you think is going to happen. You'll then test that hypothesis through experiments and collecting data, and again, making more observations. You will analyze your results using graphs, classifying. You might make models as you're analyzing those results. You finally will draw conclusions, which are going to be summarizing all of the information you've gathered up to this point, making some inferences, and modifying your experimental process to potentially retest. The final step is communicating your results, putting it out there for others to see. So we'll go through each of these steps more specifically, starting with asking questions. So scientific questions asked can find a variety of forms. Scientists might ask research-based questions like, why do bears hibernate? They may ask testable questions or investigative questions, such as, how does the amount of body fat affect the length of bear hibernation? Or they might use other questions or unsure questions like, does body fat affect bear hibernation? So as we look through asking scientific questions, we're going to focus on testable scientific questions. So a good scientific question is going to be testable. And you know that it's a testable question if it can't be Googled. So if you can Google it, it's probably not a scientific question. Phrases you might find that a scientific question or a testable question would start with might be, how can? What if? How does? What happens when? And finally, you should be able to find variables that you're testing within the question itself. So that leads us to what is a variable? So variables, just like in math, are things that are likely to change. Variables in science are things that are likely to change in an experiment. So we look at three different types of variables: independent variables, dependent variables, and controlled variables. Independent variable is the variable that is changed by the investigator. I like to think about it as I am an independent person. I control the independent variable. So an example, in the question, do plants grow taller when fertilizer is used? The independent variable here is how much fertilizer was used. So that's the thing that the scientists chose to change within their experiment, is the amount of fertilizer used from trial to trial. Dependent variables are going to be the things that depend on the independent variable. They're the things that the investigator is going to observe or measure throughout the experiment. So an example of this would be, do plants grow taller when fertilizer is used? What the investigator will measure, or the dependent variable, is how tall the plants grow based on their independent variable. So look at the different heights of the plants, considering how much fertilizer each plant had. So their dependent variable is how tall they grew. And a controlled variable is going to remain the same throughout the experiment. There can be many, many controlled variables in every experiment. For this example, we could look at, do plants grow taller when fertilizer is used? One of those controlled variable is going to be the type of plant. You wouldn't want to test different varieties of plants, like say a sunflower and a daisy, because they're clearly going to have different heights. You want to make sure that your independent variable is what is being looked at as a reason for change. So in order to do that, you have to control everything else. Other things you might control in this experiment might be the type of soil, how much water they're getting, the size of the plant, the location of the plant. So every experiment can have a variety of control variables, but it's important to keep in mind that you can only have one independent variable and one dependent variable. So why are controlled variables important? We need to know that it is the independent variable that caused the results, but also having those other variables controlled helps others know that our experiment is accurate and reliable, because they know that it's the independent variable that is potentially causing change. And it also allows for our experiment to be repeated. We want to make sure that if somebody else did our experiment, they would be able to potentially get the same results because they've controlled all the same things that we've controlled. So how do you tell which variable is which? What is being changed on purpose is going to be your independent variable. What's being measured is your dependent variable. And what's staying the same is going to be your controlled variables. So let's try this. How does the width of a rubber band affect how far it can be stretched? Pause and identify in the question the independent variable, the dependent variable, and the controlled variable. All right, let's check your work. So as we look at this, the independent variable is the thing that's being changed by the investigator, would be the width of the rubber band. The dependent variable, the thing that the investigator is measuring, is how far it can be stretched. And a control variable, in this case, might be the color of the rubber band, the material the rubber band is made out of. They can be made out of different materials. Those would just be a couple of controlled variables, but again, you could have more than one of those. The second step after we have identified our question is going to be hypothesizing. So what is a hypothesis exactly? Hypothesis is a possible explanation for an observation that can be tested by a scientific investigation. In other words, an educated guess. That's an attempt to answer a scientific question. So it's educated because we've done research on it, and that makes it not just a random guess. We've actually looked up some background information about the topic. And so we have to write this in a specific way. We follow the pattern if, and then we insert our independent variable, then, and we would insert our dependent variable, and because. So identifying something about the independent variable, identify something about the dependent variable, and then justify why. So I think the rubber band that is medium width will stretch the farthest because, and then justifying why. So let's practice. How is a flower height affected by what you can water it with? Coke or water? So the hypothesis here might be, if the flower is watered with Diet Coke or with Coke, then the flower will not grow as tall because the acid in the Coke will destroy its cells. So you've identified the independent variable, you've identified the dependent variable, and you've given a reason for why you think this is going to happen. I want you to go ahead and pause and attempt this on your own using the example in your notes, and then we'll move on to testing your hypothesis. So testing the hypothesis is the part that many people consider the fun part, but in order to get there, we have some things that we must do in order to be able to actually test our hypothesis. So testing your hypothesis, making a complete test, involves two main parts. One part of a complete procedure, or of a complete test, is a complete materials list, and the second part is a well-written procedure. So what exactly are the characteristics of a complete materials list? One is that all the materials are included, not just a few, but everything you're going to need throughout the entire experiment. Specific amounts of each of those materials are identified, specific sizes, colors, types of whatever the materials are, and that the materials are accessible. So you're gathering them prior to beginning the experiment, and that they're things that you can realistically get. You're not going to want to include things that are out of your price range or you just don't have access to. And a well-written procedure is going to have these specific characteristics. It's going to be broken down into short, easy-to-follow steps. Each step will start with an action. Pour the would be an example of that. The steps are numbered. First, number 1, 2, 3, 4. We need to have specific amounts of materials to be used included within the steps. Although you've already put the amounts in the materials list, you want to also include them in the procedure as well, so that you know how much you're using on that particular step. And then finally, you want it to be clear and concise wording. You want anyone to be able to read it and to be able to read it without it being too lengthy in order to complete. Writing a really good procedure is super important because scientists need to be able to repeat your experiment in order to reproduce the same results. If an experiment can be repeated and reproduced by other scientists, it allows the research to be credible. It gives validity to the research. Once you have your procedure written, now you're going to be able to actually conduct the experiment. So we'll continue on with observations and conducting the experiment in part 2 of the scientific method.